Prepare for our next speaker, Tom Rando from Stanford. Are you there, Tom? Can you hear me, Tom?
Yeah, can you hear me?
Yes, we can hear you, and we also see you. So you can
Okay,
just
great.
start whenever you're ready.
Okay, great. I take it you can see my slides. So thank you so much, and I'm sorry I can't be there in Copenhagen with you all. I was hoping to make it. So continuing on the theme of stem cells and aging, I'll talk today about work we've been pursuing related to a lot of different work in the lab, but focusing recently on kind of lifestyle interventions, so diet and exercise, and I'll talk mostly about diet today. So these are some of my disclosures with no conflict with the talk today. So we've been interested for a long time in systemic and tissue aging and the role in particular of stem cells in driving features of aging and ways in which stem cells can actually lead to restoration of youthful properties of tissues and organs through this kind of rejuvenation process that I'll talk about. And the stem cells that we focus on in particular are stem cells in skeletal muscle. One is shown here. These are so-called satellite cells. They're rare and they're quiescent, and they express this transcription factor, PAX7, and they're responsible really for the remarkable regenerative capacity of skeletal muscle, which is illustrated here from a normal structure through or after a total necrotic injury over the course of days and then weeks. These few stem cells, they activate out of quiescence, proliferate, differentiate, and regenerate the tissue. And it really is a remarkable feat in terms of mammalian tissue repair. But of course, like many tissues, and like we just heard in the bone marrow, that this declines with age. And this is from young and old mice with repeated injuries. You can get very good regeneration repeatedly in a young animal. But even after a single injury or after a second injury, the regenerative capacity of old stem cells declines with time. And so we've been very interested in how to restore the kind of youthful regenerative potential by targeting the stem cells themselves. And this work really dates back to the early 2000s when we pioneered the use of this heterochronic parabiotic technique to study tissue regeneration and cellular aging in particular. And we had shown through those studies that in these so-called heterochronic pairs, the exposure of the old animal to this young circulating environment really enhanced the capacity and properties and functionality, both functionally and molecularly, of the stem cells in the old tissue, so that now an injury in an old animal regenerates as effectively as in a young animal. So we looked at muscle, we looked at liver, we looked at brain, along with Tony Weiss-Corey here at Stanford. And this has been now tested in many different tissues, stem cells and non-stem cells, and shown to be a very robust process. But not just through parabiosis, but other ways people have looked at regeneration and rejuvenation of stem cells have been, I think, very illustrative. I'll just show a couple of examples. This is from, again, an older paper from our colleague Howard Chang at Stanford, in which he targeted inhibitors of NF-kappa B to the skin stem cell compartment, and showed that when he induced inhibitors of NF-kappa B, he could actually reverse some of the age-related changes, and in particular skin thickness, which is shown without the reversal and with the reversal, shown here, and shows some molecular correlates of that as well. Pen Zhang's lab at the University of Michigan had shown that treatment of mice with rapamycin was able to rejuvenate hematopoietic stem cells in this competitive transplantation assay, so that cells from old animals treated with rapamycin were really almost as effective as young cells in the transplant compared to the old cells alone. So, these are very interesting in terms of pharmacologic, genetic, parabiotic approaches to rejuvenation, but we became interested in thinking more about lifestyle interventions, and particularly diet and exercise, as a way of asking, are there ways to do this non-pharmacologically, or conversely, do these interventions lead us to new pharmacologic targets that might allow us to rejuvenate aged stem cells? So we recently published a paper asking this question: does exercise rejuvenate old muscle stem cells? And this is work by a former MD PhD student in the lab, Jamie Brett, and a current postdoc, Maria Arjona. And I won't go over this because it's published, but basically what they found was that with age, the decline in functionality of muscle stem cells is linked to a decline in cyclin D1. And that exercise can actually restore cyclin D1 levels in these cells, and through this TGF-beta SMAD3 pathway, can actually restore functionality to old cells. One of the interesting observations in that paper I just want to point out is that we found that if we took old animals and exercised them, took their plasma and injected it into non-exercised old animals, the muscle stem cells in those transplanted or transfused animals actually behave again in a much more youthful function. So it isn't so much the muscle activity of exercise, we think, rejuvenates the local muscle stem cells, but in fact, it's probably factors in the circulation that travel to those stem cell compartments, and perhaps to many other compartments that are responsible for many of the health promoting effects of exercise. So what about diet? And clearly the field of caloric restriction, dietary restriction is extremely well studied and enumerated in the aging literature, having an effect on delaying the process, slowing the process of aging. But what about the effects on stem cells? I think the literature on that is actually quite varied. And in muscle stem cells, there are a lot of conflicting data, and I think it depends on how you look at the function of these cells. And I'll talk a little bit about that. But I think a key paper is from Jennifer Morgan in the UK, did a very extensive study looking at dietary caloric restriction in different strains of mice, different sex, different times, different genetic backgrounds, et cetera. And basically, a lot of what she found was that caloric restriction, if anything, impairs the function of the stem cells in terms of colony formation, proliferation, whatever the assay might be. On some levels, that makes some sense in terms of at some point, caloric restriction is certainly going to impair cellular function. But there's been a lot of interest in ways of kind of mimicking the benefits of caloric restriction without having chronic, lifelong caloric restriction. And one of those is intermittent fasting. So we decided to explore that, like if we could understand and in a way uncouple the positive and negative effects of fasting or restriction on a stem cell compartment. And this work was spearheaded by a postdoc in the lab, Daniel Benjamin, and a PhD student, Peter Boat. And so what they did is they basically fasted mice for anywhere from 1 to 2.5 days and looked at muscle regeneration in the fasted mice. And this is just an index of regeneration. As you can see, it declines with time of fasting. And again, a little bit unsurprising given the fact that they're nutritionally deprived and it may not be as functional in that compartment. And when they looked at the muscle stem cells, they looked at the exit from quiescence and the incorporation of EDU in ad lib versus fasted mice. And as you can see, at any time point, the fasted mice, the exit from quiescence and entry into the cell cycle is delayed, markedly delayed. So just as an aside, this is how we think of this process of activation. As a stem cell, a very small stem cell goes from a single cell to 2 cells. The first thing that happens is this very prolonged growth phase, anabolic growth. And this can really take up to 2 days to get to this point, at which point the cells will enter S phase and divide. What we found with fasting is that, at least on this kind of axis, these cells take longer to enter the cell cycle as if they're in a deeper state of quiescence. And indeed, if we look at the characteristics of muscle stem cells that are quiescent compared to, say, any active proliferating cell, they have features that they're small, they have low mitochondrial content, and they have low RNA content. While in fasted animals, all of these features of quiescence are exaggerated. So the cells are smaller, they have even lower mitochondrial content, they have even lower RNA content. And they, of course, take longer to exit from quiescence. So we consider this to be, a state of deep quiescence or DQ. And that what fasting is doing is driving these cells into this deep quiescent state. So fasting has a lot of metabolic effects. So we asked, well, can we mimic this effect of fasting by mimic the main metabolic consequence of fasting, which is ketosis? So to do that, we put the mice on a ketogenic diet, so without any fasting, without nutrient deprivation. And indeed, just inducing ketosis with a ketogenic diet mimics all of these features of deep quiescence in terms of the muscle stem cell features and in terms of muscle regeneration. And we took this one step forward and said, well, what if we just replace the ketogenic diet by just injecting ketone bodies? So the 2 main ketone bodies, beta hydroxybutyrate or BHB, and acetoacetate, and we can inject them together. And indeed, injection of ketones, again, mimics the effect of fasting alone. Smaller cell size, more delayed exit from quiescence, lower mitochondrial content, et cetera, et cetera. And in fact, in the body, BHB is by far the most prominent ketone body of the 2. And basically all of these effects can be mimicked by just injecting beta hydroxybutyrate. Okay, so if the case that fasting or caloric restriction really have these benefits that we've talked about for so long in the field in terms of health span and lifespan, but this impact itself of fasting and ketosis actually impairs muscle stem cell function. It makes them less able to exit quiescence and less able to repair tissue, does fasting actually confer any benefit that we can measure on these muscle stem cells? And the answer is, indeed it does. And that is really from what we understand about quiescence. Quiescent cells are intrinsically more resilient to all kinds of extrinsic stresses. And if we use oxidative stress or genotoxic stress in vivo using irradiation, cells treated with ketones cells from animals treated with ketones or BHB show increased survival, increased resilience. We had previously published studies showing that with age, muscle stem cells are susceptible to another form of stress during mitosis called mitotic catastrophe, and the cells actually tend to die during mitosis at a much higher rate in old versus young muscle stem cells. So we looked at this in animals treated with ketones in both young and old animals, and again, there's an increase in mitotic catastrophe from young to old, but treating the cells with ketones at either young and even more dramatically at old reduces that susceptibility or increases that resilience. And we've shown that these are truly related to intrinsic cell resilience, because we can treat the cells ex vivo with BHB and again get the same kind of increased resilience, resistance to cell death, or we can take cells treated with BHB or control, transplant them, and look at the effect after transplantation. And the cells from BHB treated animals have a much higher survival and engraftment compared to control, shown graphically here. So they have higher engraftment, higher survival, and higher initial proliferation. So again, the cells are intrinsically more resilient to this kind of stress, in this case, transplantation stress. So this is the model. We have it just in a normal quiescent state. There's sort of this balance between readiness to activate and resiliency. In response to fasting or ketones, the cells lose this readiness to activate. They have a deeper state of quiescence, but they gain resiliency. They gain the ability to survive in response to the stress they're experiencing. By contrast, another form of quiescence that we've studied, which we've called G-alert, the cells are actually in a heightened state of activation, that they're more capable, more effective in regenerating tissue, but they sacrifice something. They're less resilient. So cells that are in this G-alert state tend to deplete over time, and they're more susceptible to different kinds of stresses. Okay, so I just want to, the remaining minutes, just talk about what we've been studying in terms of the mechanism by which these ketone bodies may mediate muscle stem cell deep quiescence. So we started with a shotgun proteomics approach, looking at muscle stem cells either in the quiescent state, the resilient quiescent state, or the activated state. And what we found, to our surprise, is there are quite a few metabolic enzymes that are very highly expressed in the quiescent state and downregulated when these cells enter the cell cycle. And one protein of interest is OXCT1. And this is of interest because this is, in fact, the rate-limiting enzyme in the conversion of beta-hydroxybutyrate to acetyl-CoA for entering into the Krebs cycle and generation of ATP. So we reasoned that if this is how BHB is working to enhance resilience, if we knock out OXCT1, we'll block the ability of fasting or ketone bodies to confer these effects upon muscle stem cells. So we knocked it out specifically in muscle stem cells, this gene, and to our surprise, that had no effect at all. So the cells that were knocked out had the same delay in activation, the same increase, reduction in size, same reduction in mitochondrial content, et cetera. So it seemed as if whatever we're doing by introducing beta-hydroxybutyrate, it's not by its conversion to acetyl-CoA and metabolic activity. So by what metabolic, non-metabolic activity might BHB be working? We were very intrigued by this paper from a few years ago from Eric Burden's lab, in which they found that BHB, beta-hydroxybutyrate, is an endogenous inhibitor of histone deacetylases. And indeed, in BHB-treated animals, when we look at the muscle stem cells, targets of histone deacetylase, in this case H3K9, are actually increased. So it's consistent with the idea that beta-hydroxybutyrate in these cells is acting as an inhibitor of HDACs. And indeed, if we use a fairly generic inhibitor of HDAC, gavinostat, we can recapitulate the effects of fasting or beta-hydroxybutyrate treatment. The cells enter the cell cycle more slowly, they have less mitochondria, their increased susceptibility. We can show this also ex vivo as well as in vivo. So it really does phenocopy the effects of fasting. So we did basically an HDAC inhibitor screen using a chemical library of well-known HDAC inhibitors and found that using this library and using these readouts of cell survival and cell resilience, it really appeared as if class 1 HDACs are the target. And in fact, of the class 1, it's really HDAC1 that's highly expressed in skeletal muscle. So if we look at known targets of HDAC1, here's a group of them here, we started to look at these in terms of which downstream targets of HDAC1 might be mediating this effect of deep quiescence. So we did just a pan lysine Western blot looking for proteins that might be increase acetylation in response to BHB. And we saw a few bands here in which in response to BHB, there is a large increase in acetylation. One of these bands happens to be at 53 kilodaltons. So we wondered, could this be P53? So we looked specifically at P53, and indeed, in response to fasting or ketone treatment, there's an increased acetylation of P53 in cells in these other single fiber cultures. And of course, acetylation of P53 is associated with enhanced transcriptional activity. And if we look at cells from BHB treated animals, they have a very striking P53 transcriptional signature. So we used an inhibitor of P53, Nutlin-3, and just like gavinostat, mimic the effects of fasting on cell size, mitochondrial activity, and exit from quiescence. So really mimicking this kind of deep quiescence state. So we knocked out P53 specifically in muscle stem cells. And when we did, we mostly abrogated the effects of ketone body effects on deep quiescence, resilience, and all the other features that I talked about. So this is I'll just finish up with our model. In response to fasting or a ketogenic diet, you get these higher levels of BHB in the circulation. They enter cells, and while they do go through this pathway, their effects that we're looking at here are through inhibition of HDAC1, we think, targeting P53, leading to hyperacetylation of P53, and activation of P53 downstream targets that we think are conveying this resilience. So that's the idea. And what's interesting, I think, is that we are sort of rejuvenating the cells in terms of resilience, but there's a cost. And the cost is that they have a deeper state of quiescence and a diminished activation. I think this will be a theme we'll look at a lot in terms of can we uncouple the positive effects of dietary interventions from the negative effects. And we're looking at the downstream targets of P53 now to begin to try and dissect that out. So just to end, this again, I mentioned was done by Daniel Benjamin and Peter Boat. This is pre-pandemic, and this is a post-pandemic lab meeting, both of them being very pensive at this time for some reason. They worked with other great people in the lab, both current members and former members, and we collaborated with Dan Nomura at Berkeley in the proteomics studies. And of course, I want to acknowledge the funding sources that supported this work. So I'll stop there and happy to take any questions.
Thank you so much. Great talk, Tom. Do we have any questions in the audience? Otherwise, I will start with questions from Slack, and I will choose my own question. I was wondering if fasting or ketones, if you have observed whether or not it changes or increases the time it takes to go through S phase also.
It really doesn't, in the sense that if we take the cells from animals that have been fasted or treated with ketones, we put them in culture, there's a delay in entry into cell cycle, but once they're in the cell cycle, they behave normally. So there isn't a further delay in subsequent cell cycles. It's only that activation out of quiescence.
Okay, thanks. We have a question from Aswin Magrich. Do you think muscle rejuvenation by exercise could be caused by biomechanic triggers in addition to metabolic changes?
Yeah, I mean, we certainly can't rule that out. And we've thought about testing that in a way which we have not done yet. But for example, if we exercise certain body parts and say, look at stem cells, say in the jaw muscle, which won't be specifically exercised in response to, say, wheel running or any other kind. So that's the way we've thought about getting around that, of asking, looking at non-exercise muscle in an exercise paradigm and asking if we see the same thing, but we haven't done that yet.
All right, sounds very interesting. Maybe we have a question in the audience. Someone is exercising, running, rejuvenating his muscle stem cells. Great.
Hey, yeah, Tom, great talk. I had one question. In the beginning of the talk, you alluded to cell size, and I think that's the cell size and this deep quiescence by fasting is getting smaller. And I think there's some old literature that indeed cells need to grow in order to, enter S phase. But in the end, then you focus more on P53. And do you think that the cell size has something to do with this deep quiescence? Or is it all P53? Or is that connected?
So we think it's connected. And here I can give you some sort of preliminary evidence. So in the G-alert state that I mentioned, which is the kind of sort of the opposite of deep quiescence, it's shallow quiescence. That is mediated by TOR signaling. We've shown that using genetic and pharmacologic approaches. We actually think that one of the downstream targets, or I should say pathways, there's not a direct target of P53, is again the TOR pathway. And so we think that this kind of anabolic catabolic trade-off that we're looking at here, where cells actually become smaller, is related to changes in TOR signaling. So it's P53 not acting in a genome maintenance role, but as a transcriptional pathway in a non-dividing cell that we think might be mediated by TOR. And so we think the cell size issue is highly integrally related to all the functionality that we're thinking of. And I think of these, in a very evolutionary context, they're almost like spores. They're really, if you think of evolutionary in terms of species, trade-offs between survival and reproduction. I think quiescent stem cells mimic so much of that, and that they enter this in a case of nutritional deprivation, this state of high survivability. And along with that, part of that is a reduction in size. And I think it's mediated by this P53, potentially mTOR pathway in vivo. Yeah.
All right, thank you so much.
That was Leonard, by the way, I assume I could, from the accent.
That's very interesting, super important observation. And thank you so much, Tom, for your presentation.